Good afternoon, everyone. My name is Linda Keel, and I'm the past president of Lupus Ontario. We've heard a lot of very interesting presentations so far today, focusing on actually access to your physicians, the appropriate physicians, and also the training for these physicians, as well as other barriers to care. The next presentation, I think, is very important and a great way to wrap up this afternoon, because research is one of the most important things that can be done to improve the life of lupus patients. So it's my pleasure to introduce to you Dr. Joan Wither, Senior Scientist at the Krembil Research Institute. Dr. Wither's research program is focused on identifying the genetic and immune system abnormalities that lead to systemic autoimmune rheumatic diseases, including SLE, Sjogren's, and scleroderma. So the goals of her program are to identify biomarkers for the early identification of patients at risk for developing systemic autoimmune rheumatic diseases, or SARD, to evaluate the therapies for prevention of SARD, to characterize the immune mechanisms that lead to disease flare and progression. And this information will be critical to improve diagnosis of complications and tailoring treatments to each patient's disease, and to develop diagnostic tests using novel biomarkers to monitor progression and severity of SARD complications and response to therapy. So we're very fortunate to have Dr. Wither talk to us today about interferon's role in promoting lupus flares by modifying B-cell function. So welcome, Dr. Wither, and over to you.
Thank you for the opportunity to tell you about our work on interferons and how they act to promote disease activity in lupus. These are my disclosures. So interferons have been known to be elevated in lupus for many, many years, but it wasn't until approximately 20 years ago that it was discovered that the vast majority of lupus patients actually have elevated levels of interferon. And this was discovered when a new technique was developed to look at the blood cells of patients with lupus, where they were able to look at the expression of the genes in the blood cells, and they were able to compare genes that were expressed in lupus patient cells versus healthy control cells. And it was appreciated that 70 to 90% of lupus patients actually had increased levels of genes that shared the property that they were induced by interferon. And actually you can see, oh yeah, here we go. You can see this red patch here that is in the lupus patients, and at the front and back are controls, and these are the interferon-induced genes. So this actually has led to the name of an interferon signature, meaning that lupus patients, many lupus patients, have this change in their blood cells that suggests that the blood cells have contacted interferon. So what are interferons? Well, interferons are proteins that are secreted in response to viral infections, and they're critical for fighting infections such as COVID-19. There are several different types of interferons, which are shown on the right here. All of them are capable of inducing interferon-induced genes, and they bind to receptors on the surface of the cells that induce expression of these genes. And these genes normally respond to interferon and allow us to facilitate the response to viruses and help us to clear viruses. So interferons have many effects on many different cell populations. I'm focusing on B cells today, but they can affect many other cell populations in the blood. And basically they act on these immune cells to arm them to fight infections, specifically viral infections. So what causes interferons to be produced in lupus? So it turns out that there's a special cell that produces interferons in response to infection, and that cell also produces interferon in lupus patients. And it's been shown that in lupus patients, there are autoantibodies against many nuclear proteins, the so-called anti-nuclear antibodies. And these anti-nuclear antibodies can be taken up by these plasmacytoid dendritic cells. They're complex with nuclear antigens, and these induce the plasmacytoid dendritic cells to produce high levels of interferon. So production of interferons very closely associated with the production of autoantibodies in lupus. So does interferon promote development of lupus? Maybe it's just an epiphenomena. Well, it's actually been known for a number of years that if you take patients and treat them with interferon alpha, which is being done in patients with hepatitis C or interferon beta for multiple sclerosis, that you can actually, in a very small subset of patients, induce development of lupus. And usually when you stop the interferon, the lupus will resolve. In addition, it's known that if you look at patients before they actually develop a lupus, many years before they have autoantibodies, and if you track patients over time, those patients that have the highest levels of interferon tend to be those patients that go on to develop lupus. So it seems to be playing a pathogenic role in lupus. So what clinical features are associated with the production of interferon in lupus patients? So most studies have just studied lupus patients at a single point in time, and most studies have used a cluster of genes induced by interferon to measure the so-called interferon signature. And they have shown that at a single point in time, the levels of interferons in the peripheral blood, which can vary very much from patient to patient, are correlated with disease activity, especially active disease in the skin, the muscles, or the joints. So this led to a lot of interest early on in interferon as a potential biomarker for flares of disease in lupus. So the question was, does interferon go up and down with disease activity, and could it be used to predict impending flares? So a number of years ago, we actually showed that the interferon signature does not fluctuate in any predictable way with disease activity when you follow a patient over a prolonged period of time. And then subsequently, we actually did look at a number of patients, and we measured their interferon signature or interferon-induced gene expression using various clusters of genes over a period of about a year to 2 years. And what we could show, as shown by these black lines here, which are two types of interferon-induced gene expression, that the levels of interferon-induced genes are stable over time despite marked changes in disease activity. For example, this is disease activity as measured by the SLEDAI, and you can see this patient was active and then became quiescent. Or here, this patient was inactive and became active, yet there was no major change in the interferon-induced gene expression. And we have now looked at this, and others have looked over this over even more prolonged period of time, like 4 or 5 years, and not seen major fluctuations in the vast majority of patients in interferon-induced gene expression. So this raises the question, why in cross-sectional studies, when we look at a patient at a single point in time, does the interferon signature correlate with disease activity, yet it doesn't go up and down with flares? So a clue to this came when we went back and looked at our original study about 10 or 15 years ago now, and we found that patients who had high scores, interferon gene scores, had more recently had a flare. So this led us to question whether there was an association with disease activity simply because patients with higher interferon scores had more severe disease. So they spend more of their time in active disease. So to look at this, we measured the interferon score in 137 lupus patients that we had followed for at least 5 years. And we assessed the correlation between various measures that reflect increased disease severity over the subsequent 5 years. And shown in this slide is our findings. We found that on the horizontal axis, this is increasing interferon score or increasing levels of interferon as you move to the right. And on the vertical axis, the various measures of disease activity or disease severity. And we found that the higher your interferon score, the higher your average disease activity over the subsequent 5 years. The more frequent flares you had over the last 5 years. And that patients with high interference actually required a higher dose on average of prednisone over the subsequent 5 years as compared to those with low levels of interferon. In addition, patients with high levels of interferon required more changes of immunosuppressive medications because of difficulties getting them under control. So this led us to think that interferon was associated with disease severity. Now, we also looked at the time to flare and frequency of flares, which I showed you previously, but the time to flare in patients, and we compared patients with high levels of interferon with those with low levels of interferon. And what we found was that patients who had high levels of interferon had a shorter time to flare and had more flares subsequently than patients with low levels of interferon. And this was true whether or not you were active at baseline or whether you were inactive at baseline. So in summary, what I've shown you so far is that high levels of interferons and interferon-induced gene expression are seen in the majority of patients with lupus, that they are stable over time. And this is likely because the antibodies that induce these levels of interferon are also stable over time. They tend to be what we call long-lived antibodies. And finally, I've shown you that high levels of interferon-induced gene expression are associated with more severe disease. So the next set of slides are on how interferon actually might act to promote the development of flares and more severe disease. So it's known that autoantibodies, those are antibodies that bind to the patient's own self molecules, play a very important role in lupus. These antibodies, which are typically anti-nuclear antibodies that bind to DNA, RNA, and proteins that are complexed with them, form what's called immune complexes. They bind to these self molecules in the blood, or they bind to them in the organs, and they cause inflammation there, which causes disease activity. Now, antibodies are normally produced by B cells in a typical immune response to help clear infection. And the production of autoantibodies is actually actively suppressed and prevented in healthy individuals. And the way this is done is actually by quite a number of immune mechanisms, which collectively are called B cell tolerance mechanisms. So the question we asked was, do high levels of interferon actually disturb these B cell tolerance mechanisms and actually promote the production of autoantibodies? Now, to do this, we looked at a mouse model. So I am going to get back to humans, but for this initial experiment, we developed a mouse model. And what we tried to create was a mouse that had some changes that were associated with lupus. And the way we did this was taking normal healthy mice and genetically modifying them so that a proportion of their B cells were recognizing self-DNA. So just like in lupus patients, where lupus patients have B cells that recognize DNA and that go on to eventually produce autoantibodies against DNA, we modified these mice so that these kind of cells were enriched in their B cell population. So normally it was shown that these mice do not produce autoantibodies against double-stranded DNA or single-stranded DNA. And the reason is that all these B cell tolerance mechanisms that prevent reactivity to self prevent these B cells from making antibodies to DNA that could potentially cause some damage. So we took these mice that were otherwise healthy, except for this enrichment of B cells that recognize DNA, and we infected them with a virus that produced sustained high levels of interferon to mimic what happens in lupus patients. Now, we also genetically modified these mice, or a subset of these mice, so that the B cells specifically lacked the receptor that bound to interferon. And this allowed us to determine whether the interferon was causing abnormalities because it was directly affecting B cell function, or whether it was causing these abnormalities because it was affecting other cells that might be affecting B cell function. So what we found was that just taking this healthy mouse and giving it sustained high levels of interferon was sufficient to overcome B cell tolerance mechanisms and lead to production of anti-DNA antibodies. So that's actually shown here, where what I've done is drawn a red box. In this first column here is the mice that received interferon alpha, and the second column is the genetically modified same mice that lack the receptor on B cells. So what this shows is that interferon acts directly on B cells to modify their function and to allow them to differentiate into antibody-producing cells that bind to double-stranded DNA or single-stranded DNA. So now I'm going to digress a little bit, because it's known in humans which cells are the precursors of the antibody-producing cells that bind to single-stranded DNA and double-stranded DNA. And those cells are called ABCs, for age-associated B cells. Now, I'm not going to get into why they're called age-associated B cells, but it's known that ABCs are the immediate precursor of the cells that secrete these autoantibodies in lupus. So, now what's happened here? There. So here's a picture of an age-associated B cells. And what was known is that these B cells are associated with high levels of interferon. So people with high levels of interferon tend to have more of these B cells than people with lower levels, and associated with high disease activity. And as I mentioned, they're the immediate precursor of autoantibody secreting cells. And what you can see on the left is these are the experiments in our mice. And this column right here is those mice that received interferon, and they have high levels of ABCs as compared to mice that didn't receive interferon. And this was a direct effect on these B cells, because when we knocked out the receptor for interferon, there were no longer ABCs. So this suggested that interferon acts directly on B cells to promote differentiation of these ABCs and autoantibody secreting cells, leading to the production of anti-DNA antibodies. And high levels of interferon are sufficient to do this. So I'm not going to go over every mechanism of B cell tolerance, that is disrupted by high levels of interferon. This is the graphical abstract from the paper that we recently published. And basically, what it shows is that every single mechanism of B cell tolerance, every single mechanism that is invoked in the immune system to prevent self-reactive B cells from getting into the mature B cell repertoire, from developing into antibody secreting cells, etc., is disrupted by interferon, so that it has a broad-based effect on B cells, causing them to make autoantibodies. So the next question that arose is, this experiment was done in mice. Does the same thing actually happen in humans with lupus? Are those ABCs actually very important cells? So to address this question, what we did was to look at what are the immune changes that are associated with flare in lupus. Are there specific changes in the immune system that are associated with flare or not? So to do this, we looked at 46 patients with a recent flare, which we defined as being a change in disease activity over the last month that led the physician to change their therapy. And then we compared them with 25 patients who had sustained clinical inactivity over the past year. So these are patients that were not active. They had no increase in prednisone over the past year, no change in immunosuppressive, and they were on less than 10 milligrams of prednisone. And we set it at 1 year because we thought maybe the changes that were associated with flare maybe wouldn't change that quickly following the flare. That even though patients are treated and they get better, maybe the immune changes would still be there. And we followed these patients over time, and we measured 47 different immune populations in the blood. Now, I know this sounds like it's going to be complex, but it's not. So I want to focus your attention on cluster 1, which is the far left here. And I've drawn 2 red boxes over the distinctive changes, in that cluster of patients. Okay, so what you do is you measure all these different cellular populations. And then using machine learning, and you can call it AI too, you get the computer to cluster the patients into those patients that are similar to others from their cellular perspective. So when you do that, you end up with this group on the far left, which is called cluster 1. And if you look at the red bar that says status, the bar here that says status, what you can see is a lot of the red patients, which are flaring patients, are in this cluster. And the pink are flaring patients that follow up. And there's lots of pink patients in this cluster too. So, and if you look at the SLEDAI overall, the SLEDAIs are pretty high in this cluster compared to some of the other clusters. There are no healthy controls in this cluster. The healthy controls are here on the far right. So what's distinctive about this cluster? Well, they have those ABCs. They're expanded in that cluster. And they also have lots of B cell activation, which is one of the things we saw in our mice that received high levels of interferon. So this cluster is unique in that it has a high proportion of ABCs. These patients have lots of B cell activation. They have high levels of interferon-induced gene expression globally in their peripheral blood. They have a high proportion of active patients. And when we looked at prior to these patients being recruited and 1 year after this blood was drawn, what we found is that the majority of the patients in this cluster were still active at 6 months and 1 year. And this cluster of patients was most likely to have flared in the previous year before they were developed this flare. So I think overall we can say that this cluster of patients has more severe disease as compared to all of the other clusters. So high levels of interferon, and as we saw in mice with high levels of interferon, high levels of ABCs and high levels of B cell activation. So do the ABCs actually in patients with lupus have high levels of interferon-induced gene expression? Well, it's very hard to measure interferon-induced gene expression on a single cell level or a single population level. You can do it, but A, it's expensive, and B, it's technically challenging. So what we decided to do was, instead of measuring interferon-induced gene expression on a single cell level, we thought, why don't we just use flow cytometry, where you look at single cells all the time? But instead of looking at gene expression, we'll look at proteins that we can actually easily measure using this approach. So what we developed was a CyTOF panel that enabled examination of interferon-induced protein expression. And we use this as a surrogate marker of interferon-induced gene expression, because when the genes are expressed, usually that leads to increased expression of the proteins. And this allowed us to look at all kinds of different cell populations and see, were they exposed to interferon, and or were they all exposed in the same way, or do some cells get exposed differently than other cells? So I'm circling here the B cells populations at the top right of this panel. Every one of these blobs is a different cell population. So this blob, for example, is something called monocytes. And in here we have the T cells. So we have different cell populations. Every blob is a different cell population, but the B cell populations are all in these blobs here at the top. Now, low levels of interferon-induced protein expressions are blue, and high levels go through the yellow spectrum to get to red. And so what you can see is, here is a healthy control. They have low levels of interferon in their B cells. This is a quiescent patient. Lupus patient has slightly higher levels. And here is the flaring patient, which has the highest levels of interferon-induced protein expression in these cells. And what we found was that actually every cell in the lupus patient, if they had high levels of interferon-induced genes in their peripheral blood, had high levels of interferon-induced proteins in every single cell population in their peripheral blood. So here are pre-ABCs and here are the ABCs. And what we found was there was an excellent correlation between these interferon-induced proteins and the interferon-induced gene expression in the peripheral blood. So they've all been exposed to interferon, likely in the blood, not just in one little part of somebody's body, but in the blood. And they all have a very high levels of these interferon-induced proteins, which correlates with the levels of interferon-induced gene expression. So this is my final slide. And so we asked the question, we know that ABCs were associated with more severe disease. We asked whether there was anything that predicted how active you were going to be 1 year after your treatment, and what predicted 1 year how active you were going to be after treatment or no treatment. If you were quiescent at baseline, was your interferon-induced protein expression, in this case, in your ABCs. So again, this suggests that interferon A is probably driving ABCs, and B, that it's important in terms of disease outcomes. So what I've said in this part of the talk is, patients with increased levels of ABCs have more severe disease. That the levels of interferon-induced genes, or in our case, proteins, in ABCs reflect those seen in the blood as a whole. So measuring interferon-induced gene expression in the blood as a whole is a pretty good biomarker, I think. And as I said, high levels are associated with high interferon levels. I didn't show you that, but we have looked at that, and that is the case. High levels of interferon-induced protein expression in ABCs predict ongoing disease activity at 1 year despite treatment. And this suggests that one of the mechanisms by which interferons induce disease activity is by driving B cells down this ABC pathway, causing them to differentiate and produce high levels of autoantibodies. And we have done some cultures where we've taken normal, healthy cells and induced their differentiation to ABCs, and we show that interferon, our preliminary data suggests that interferon actually enhances and promotes this. So that kind of finishes the loop, sort of showing that what we saw in our mice is applicable to patients with lupus. So I'd like to acknowledge all the people in my lab who work hard and have done all these experiments. I've tried to show you pictures of some of them as we went along. You know, all of this work was done in collaboration with the University of Toronto Lupus Clinic. And so there are the collaborating clinicians that I think many of you know very well. We also have done some work with people at PMH. The person who originally developed the interferon signature actually for use in cancer patients with these interferon-induced proteins by CyTOF was David Brooks. And so we've adapted his work. He's our collaborator on that experiment so that we can use it to look at the peripheral blood in lupus patients. And Giselle is a biostatistician who has helped us with a lot of our data analysis. And at the bottom are our funders. And I would like to highlight Lupus Canada because Lupus Canada funded our original CyTOF experiment, that one where we were looking at interferon-induced proteins in individual cells. And if they hadn't funded us, I don't think we would have been able to look at that. So and also I think that also they just funded us in another experiment where we're looking at interferon-induced proteins in the kidneys of patients and seeing whether that can predict treatment outcomes. And then the Arthritis Society, CHR, and the UHN Foundation have also funded us. So thanks for listening, and I'm open to any questions or comments.
Thank you, Dr. Wither. That was very interesting. We have a couple of questions in the Q&A. And the first one is why interferons blocker is not working for lupus nephritis.
Well, no, I don't think we know that for sure because that study is still in process. You know, when they did the first two studies with anifrolumab, I'm assuming you're talking about that, they specifically excluded lupus nephritis patients from those studies. So they weren't able to comment on lupus nephritis. I think there's an ongoing study of lupus nephritis. I do not know that the outcome of that study is being released yet. But blocking interferon certainly does work, at least from the published studies. It does decrease the severity of disease activity. It's been shown to lead to a low disease activity state, which in other studies has been shown to be associated with the decreased likelihood of damage and a better outcome over time. And it's been shown to reduce flares of disease activity. So the converse of what I showed for a high interferon signature appears to be true when you block interferon with anifrolumab.
Okay, I saw a note come up on the Q&A. Brent, did you want to answer that question or did you have a comment on it?
Nope. I was just marking that we were going to answer it live. We did that. So that's what I was marking. I have a question. I'll answer or I'll ask it later on. I think there's still one more in the chat.
Yes, there is. Yes. Is there any difference between peripheral and intrarenal B cell depletion between B cell depletion therapy?
So, okay, the B cell depletion therapy is less effective in the tissues as compared to the peripheral blood. So it's known that sometimes we think that we have depleted B cells, but in fact, it's not as effective in the tissues as in the blood, and we may not have completely depleted B cells, and that might be why some patients fail treatment with B cell depletion. I mean, there's a really nice study, which I'm sure you're all aware of, with the CAR T cells, where they were very effective at depleting B cells and likely some the plasma cells, so the cell antibody producing cells in lupus patients. And that was a very effective means of treating lupus. And to me, it shows that if you can get rid of the B cells and the autoantibodies, you can treat lupus. It just shows that the B cells and autoantibodies are very important in lupus. If you get rid of them, you can treat lupus. There are some wrinkles to that treatment, but so, up to that point, we had thought that getting rid of the B cells was not always effective, but it does actually show if you can really effectively deplete B cells that you can treat and their how do I say, and their consequences, the plasma cells or antibody secreting cells, you can treat lupus. So it gives us hope that if we can develop more targeted therapies, that we will be able to treat lupus very effectively.
Well, that is very encouraging news because we're all looking for better treatments. Brent and Hadra, I don't see any more questions in the Zoom. Is there anything on FeedLoop that we should be addressing, or are they one and the same?
I don't see any more questions. Anne, do you see any at all?
There are no additional questions through FeedLoop.
So I have one, and Dr. Wither, I think you actually spoke to it, but I'm going to ask the question anyway. And I'll do it by way of analogy to a degree. I know, so in multiple sclerosis, for example, there have been recent studies that showed that individuals with MS, by and large, had contracted the Epstein-Barr virus. So the analogy was, if you had MS, likely you had been infected with the Epstein-Barr virus, but having come in contact with the Epstein-Barr virus did not necessarily mean you were going to contract MS. So my question is by way of analogy here. If you have these high degree of interferons, does that automatically mean that you're going to contract lupus or have lupus, or you're going through a flare? Or is there some sort of nuance there that if you have what you've presented here with the interferons, you're not necessarily going through lupus or have lupus?
Yeah, so one of our arms of research is looking at autoantibody positive patients and seeing what predicts those patients that have autoantibodies, because as you know, anti-nuclear antibodies are very prevalent in women, with about 20% of women having positive anti-nuclear antibodies. And we've shown that about 30% of those women will have high interferon. So having high interferon by itself, does that predict? I do not think that everybody who has high interferons is going to progress. having very high levels of interferon certainly does enrich for progressors, but even having very high levels of interferon, we have a number of individuals with that, and they have not as yet progressed. We have shown that combined with a couple of other biomarkers, that there is an increased, fairly high rate of progression, but they indicate that it's not, maybe not sufficient just to have high levels of interferon. So that's what I would say.
Perfect.
Does that make sense?
Yes, yes. Thank you very much. We do have one more question in the chat now. We only have a couple of minutes left, though. So I'll ask it, and then maybe we can do a 30 second, 60-second kind of quick hit on it if possible. The question is, what is the effect of ESR test in lupus patients?
Oh, I'm not sure I'm going to be able to answer that. You know, many things induce elevated levels of the ESR test. And not always lupus. For example, any kind of infection you get can increase your ESR. And I do have to say that I do see some preclinical patients that have high ESRs. I have not found to date that is predictive. That's all I can say. Generally, it's supposed to indicate more inflammation. But I do not know of any study that shows that the ESR going up and down is a good mirror of flares. You know, in fact, the things that mirror flares are low C3 and high double-stranded DNA, but not in everyone. So I am not sure of the importance of ESR as a biomarker of disease activity.
Thank you very much. So ladies and gentlemen, that will do it for today's presentations. We have only about a minute and a half left or so. So with that time, Dr. Wither, I'd like to thank you very much for your time, for joining us here today and this afternoon. This has been an incredible presentation. And I know I have more questions. We can save that for another rainy day. But thank you very much for your time. I'd like to also thank Anne and Hadra in the background who were keeping us running and really were the hostesses for our event today. And I'd like to also point out to everybody that we do have an exhibitors portion in our portal. If you look under the directory tab, you can see individual exhibitors there and look for information, as well as an interesting study on COVID in individuals with immunodeficiencies or immuno issues, in particular lupus. So you may want to have a look at that if you're interested in COVID and getting involved in some research there. I'd also like to invite everybody to a webinar on November 27th at 7 p.m. with Dr. Celios speaking on biologics. So we're just about out of time, but again, thank you everybody very much for joining us today, and Dr. Wither and all of our speakers as well. Thank you, and we'll do this again hopefully next year as well. So in the meantime, look for new webinars and have a wonderful weekend.
Thanks. Thank you.